and welcome to ZOOMinar. First, let me go through several updates and announcements for the ZOOMinar series. With the hope of returning back to normalcy by the end of summer, this ZOOMinar series is now scheduled to continue until the end of August this year. We're also hoping to have an on-site conference in Sicily, Catania, Italy, sometime in September or October. This event was scheduled to happen last year, but it was canceled due to the pandemic. We'll bring you more details in the next couple of weeks or so. We're pleased to inform you that we'll have two mini symposia scheduled for June 23rd and July 14th. It's about 4 hours or so, 9 to 12 in the morning, Michigan time. We'll hear the presentations from early career researchers on topics in the area of proteinopathies. I take this opportunity to thank the committee for doing a great job in finalizing the candidates for live presentations for those two mini symposia. Martin Michel, the chair of the committee, Ifad Miller, Sandrine Wanjeri, and Zhuang Wang, Anirban Bunia, Michelle Siaka, and Magda Ivanova. Thank you very much, guys, for the great job. I really appreciate it very much. Now we would like to bring another opportunity for the early career researchers through the Next Generation Symposium organized by Dr. Dylan Murray from UC Davis. Dylan will tell us more about it. Dylan, go ahead.
Okay, thank you. Yeah, so hello, everyone. I'd like to invite you on behalf of the organizing committee, which is not just myself, to the Next Generation Symposium of the FASIB Protein Aggregation Conference. It takes place the day before the conference on June 2nd, 2021. Of course, it's going to be virtual. The cost is free. It's intended to give postdocs and graduate students opportunities for talks, but we'd like to extend the invitation to attend to scientists of all career stages. I'm going to put a link in the chat to the conference website. It's on the main FASIB Protein Aggregation conference website under resources. And there's a Google form you can fill out, submit an abstract. The deadline is actually May 24th to give priority for those giving talks, but if we will accept abstracts up until the day before the conference, 2 days before the conference. Yeah, so thanks for the opportunity to advertise our symposium, and I'll put the link in the chat. Thank you.
Thanks very much, Dylan. So today we have two presentations. The speakers are Dr. Van der Wel from the University of Groningen, Netherlands, and the second speaker is Dr. Gareth Morgan from Boston Medical School. I also would like to welcome the guests and moderators to the panel, Dr. Antoinette Killian from Utrecht and Dr. Wolga Gursky from Medical School of Boston Medical School. So with that, I would like to ask Antoinette to get started.
Yeah. Thank you, Rens. It is my great pleasure to introduce the first speaker, my fellow countryman, Patrick van der Wel from Groningen University in the Netherlands. So Patrick's expertise is the use and development of advanced solid-state NMR spectroscopy techniques, which he uses in particular to study protein-lipid complexes, oligomeric chaperones, and aggregated proteins. And he focuses not only on the structure, but also, or perhaps I should say particularly, on the dynamics and the molecular interactions. So a little bit of background. Patrick studied chemistry at Utrecht University, where as part of his studies he joined my group. And this was around 1995-96. And I had just started my own independent group then. So Patrick and I go a long way back, and I really have good memories from that time. So it is at Utrecht University that Patrick first became acquainted with NMR, with membranes and with peptide-lipid interactions. He then went to the United States for a PhD with Roger Cuppy at the University of Arkansas, looking further into peptide-lipid interactions with solid-state NMR approaches. And we do have a number of joint publications from that time that I'm very happy with. So Patrick then further specialized in solid-state NMR methods and method development during a 5-year postdoc at MIT, working with Bob Griffin. And at that time, of course, he also became familiar with and interested in amyloid-forming peptides and proteins. After that, in 2008, he moved to the University of Pittsburgh, and there he obtained a tenured position as an independent group leader. He focused further on protein misfolding and protein-lipid interactions, with as a central focus the structural biology of protein aggregation in Huntington's disease. And as you can see, this is the title of his talk, and in related polyglutamine disorders. So I was really happy that in 2018, Patrick decided to return to the Netherlands, where he was offered a faculty position at the Groningen University. So he presently leads the solid state NMR group of the Zernike Institute for Advanced Materials. And this means he is also further broadening his research to other types of materials. But today the focus will of course be on protein aggregation. So Patrick, I'll give the word to you now, and I look forward to your talk.
Well, thank you, Antoinette, and very nice to such a nice and historical introduction. And indeed, yeah, like it says on my slide, and I'm now in the Netherlands in the University of Groningen. But until recently, I was in Pittsburgh, and most of the work that I'll talk about is done still in Pittsburgh by the team that I was leading there then. But some of the a lot of this work continues now in the Netherlands. I hope that the slide just move. I mean, I mentioned before that I've had trouble.
That's good.
Okay. So yeah, so we are getting a bit spread out now among various topics, where we, on the one hand, are getting more into some material science topics with some of my colleagues at the Material Science Institute in the University of Groningen. But more traditionally, my work has been more on using solid-state NMR to probe biological membranes, as well as protein aggregation processes. And one of the key topics that we've sort of by chance specialized in the membrane field is cardiolipin and mitochondrial protein interactions. And we're just in the process of publishing a paper in which we actually cite some of Antoinette's work on the same topics from many years ago. So it's a kind of a circle, small world kind of thing here. But during my time in Pittsburgh, I really got started in studying these polyglutamine proteins and polyglutamine aggregation. And that's really what I'll focus on today, which of course has led to some side projects, including a nice paper that we have recently published in Nature Communications on a chaperone that counteracts PolyQ aggregation. But today I'll focus a bit actually more on some work that has been published on PolyQ aggregation. And so the key example of aggregation of polyglutamine proteins is in the most common disease that's associated with it, which is Huntington's disease. And it's a fatal inherited disease where the patients suffer from motor disorders and brain functional problems, like listed in summary on the slide here. And interesting feature of this disease is that people, of course, are born with the mutant protein, but it only manifests itself upon aging, and it happens during adulthood usually. But on this point of time, there is no prevention and no cure for this disease. And the disease is so bad that many people even choose not to get genetically tested to know, because they don't want to deal with the prospect of knowing what's coming for them. And so there's a lot of work going on now to try and find ways to counteract this disease. And part of my work is really driven by the idea that we can contribute to understanding some of the molecular mechanisms involved to maybe participate and contribute to both treatments. And one of the things we're working on now is also to help understand how we could have diagnostic tools to see the aggregation of proteins in the disease. But I'm kind of jumping ahead here. When you look at patients that suffer from this disease, you see that they have clear brain deterioration. You can see this for a control and a patient, even pre-symptomatic. You already see dramatic changes in the brain matter. And if you look more closely, what people discovered in the 1990s is that you see that there are protein inclusions in and near these areas of the brain. And that if you look inside these inclusions, you find that there's a particular protein involved, this Huntington protein, which is mutated in these patients. And I cite here a particularly nice review from a couple of years ago now that really is a great primer into understanding this disease. So the Huntington protein itself is a pretty large protein, at least from a structural biology point of view, more than 3,000 amino acids. And until recently, I would have said it's been refractive to structural biology efforts. The mutation happens in the very beginning of the protein, only in the first exon of this large protein. And what you already see in this older model is that you see a lot of wiggly lines, disordered regions. And so this is more than 3,000 amino acids long. Now, what has happened in recent years, as many of you probably realize, is that you see cryo-EM really making huge breakthroughs in solving complicated and really impressive protein structures. And in the case of Huntington, this has also been true, where people solved Huntington partially based on the advance in EM and partially by binding a not pictured here protein that stabilizes the structure of Huntington. So you get this protein structure that's been resolved, and people get really excited that we can now learn something about Huntington's disease, perhaps up until the point where you realize that the first part of the protein is actually invisible in the structure because it's disordered. So it's an example of a disordered segment in the protein, so an IDP or IDR. And so we actually don't really know from this structure what the Huntington fragment looks like that's affected by, but it's relevant for the disease. And then if we zoom in on this sequence of this first exon, you see that it features this segment of glutamines, a glutamine repeat element. And it's this segment that it gets expanded or lengthened in the patients. And so what you see is that for most of us, hopefully, we have around 20 glutamines in this segment in our Huntington proteins. But in patients, you see that the Q length increases to 40 or beyond. So so clearly, somehow, this mutation is what drives the disease, and this makes it into, to a large extent, a monogenetic and sort of predictable disorder. And what I'll talk about later is that these regions surrounding this PolyQ segment are also relevant and interesting to understand, and we'll see that more later. But the Q segment here is only 17 residues from the very end terminus of the protein. Now, if you look at this number of CAG repeats versus the age of onset, you see a pretty dramatic trend where the longer the Q length is, the more early people get affected by the disease. And what you see in this figure also is that there's a whole bunch of diseases that all have PolyQ segments, which all show this dependence or this correlation between age of onset and PolyQ length. But there are some interesting differences between these diseases. And so far in our work, we have focused on this Huntington disease case. And then this figure here on the right shows that people have shown that PolyQ segments themselves have a high aggregation propensity, even though if you look at the chemical structure of glutamines, you may not initially think so. And this also leads me to introduce the fact that this is work by Ronald Wetzel, which many of you know, I think, in the field. And he's the one who really talked me into starting to work on PolyQ aggregation. And when I started my lab in Pittsburgh, he sat in the office next to mine, really allowing us to work together very closely on tackling this interesting but challenging system. Now, one of the things people discovered quite early on, also in the 1990s still, is that this first exon is actually seemingly sufficient to get some sort of disorder in mice, if you put it into mice, and it's now been transplanted into all sorts of other model animals. This leads to the idea that this exon 1 is maybe a particularly relevant segment to study for the disease. And what I also mentioned, I think, but maybe skipped over a bit, is that the aggregates people see in patients also appear to contain N-terminal fragments of this large protein complex. So in the fibers here, we do not have the entire protein, but we only have the first segment of the protein. So if you take this exon 1 segment, then you make it in its expanded form, so with 44 glutamines, like a Huntington patient would have, you see that it over time has the propensity to form kind of oligomeric species and ultimately also fibrillar species. And these fibers then ultimately mature into a morphology like this. And this happens to be our EM data, but other labs, many labs have now shown, or have before and after us shown the very similar morphologies. Characterized, what you can't see very clearly here, there's sort of a fuzzy coat type architecture on the surface of these fibrils as well. Now, some really cool work by a bunch of people in Germany have shown that you can also do tomography, EM tomography, on these kind of aggregates in a cellular context. So the aggregates formed inside an actual human cell. And you get these large inclusions, but if you zoom inside the inclusions, you see that they're made up of individual filaments, and these filaments, at least by the statements of these EM spectroscopists, resemble those seen in vitro. And they also correlated to the kind of structures reported by my group here, but also the group of Ansgar Siemer and Ralf Langen at USC, who have also extensively studied these kind of aggregation processes with both NMR and EPR studies. But they also note that so far it's been really tough to tackle these fibers with high resolution EM because of their heterogeneity. And I'll talk a bit later about how we see that in our NMR data. So now to go back to the kind of biology here, one of the reasons that we're interested in studying the structure of these aggregates, these protein inclusions, is that we really think that they contribute in some way to the disease, and that understanding their structure, but also how they form, can help us find ways for diagnosing the disease, but also for understanding ways to interfere with or to understand or explain the toxicity seen in neurons. And I kind of wanted to comment on a couple of aspects of this. Is that you could imagine, or we know that these kind of fibers interact with membranes inside the cell. In this prior study, they've shown that it interacts with the membranes of the Golgi and mitochondria, for example. We also know that once formed, these fibrils will sequester and attract and trap other variety of proteins, chaperones, PolyQ proteins, and other types of proteins, and thereby eliminate function from the cell, but themselves thereby becoming effectively toxic. But of course, there's also the idea that fragments of these fibers, or oligomers, could themselves act as toxic species by punching holes in membranes and those kind of things. And of course, there's also the idea that these kind of protein clumps can interact with transporter proteins or receptors, thereby either blocking essential function or by causing the misdirection, the misfiring of certain cellular processes. So what we are setting out to do then, or what the lab has been working on in all these years, is to see, can we use solid state NMR to actually probe the atomic level structure that's present in these fibers? Then can we use that information to think about Huntington's disease pathogenic mechanisms, and in particular, what might drive or dictate disease onset? And so what I've already shown you, those images of our fibers, show that we can make these proteins in the lab and then have them aggregate and then do NMR spectroscopy on them. I wanted to take a short moment to point out that we attempt to look at these fibers suspended in a solution, in a buffer, and use sedimentation or pelleting to generate hydrated fibrils that we study by NMR. So we're not looking at a dry powder, but at a hydrated protein, which allows certain parts of it to remain its flexibility that they would have in a biological context. Then ultimately we get by NMR spectroscopy 2D and in some cases 3D NMR spectra, but we actually use also quite a fair bit of classic 1D NMR to probe the behavior, dynamics, and structure of these proteins. And then the interesting thing with these PolyQ aggregates that we see in a lot of work on amyloid fibers with solid state NMR, including the work by Dylan, who is we just heard from, is that you see that only a small part of the protein is actually forming a kind of rigid structure in these aggregates. And that's done with this sort of canonical or typical cross-polarization based NMR spectroscopy, where we see only a subset of the residues that we would expect to see if you look at the sequence of the whole exon 1. And this whole statement is something you can apply to many different types of fibers. And so what that means is that a lot of the peaks that we would expect to see are missing because those residues happen to be flexible or dynamic. Another key feature that I should point out here, and I've touched upon briefly before, is that in our protein here, we don't just have the PolyQ segment alone. We also have the N-terminus, this N17 segment, and the proline-rich C-terminal domain that flank this PolyQ domain. And I can't really talk about that here due to time constraints, but we've shown and studied how these domains actually greatly accelerate or slow down aggregation by the PolyQ domain. But in our NMR data, what we see is that we see some signals from this N17, the Qs, and part of the proline domain as part of these kind of solid NMR signals. And a bunch of stuff here at the end is actually missing in these kind of spectra, showing that these are flexible parts. Now, if we zoom in or look at this PolyQ segment itself, which of course is really interesting because it's the mutation site, then we can pinpoint that there are two types of signals showing up. So you can see this here in this part of the spectrum. There's the kind of peak that are marked with red and the peak marked with blue lines. And we identify these are two different conformers, two different conformations of the glutamines. And it's interesting because we know that this domain has actually 44 of these glutamines. So we don't see 44 peaks, we see just 2. So these 44 glutamines somehow adopt 2 different types of conformations. And we can do NMR to show that actually these 2 conformations are interacting with each other in the fibers. So we see polarization exchange between them. So they're somehow co-assembling in the fiber core. And we've spent a bunch of time trying to analyze this, but what you quickly realize is that if each peak has something like at least 22 glutamines in it, you can't really use the canonical way of identifying distances between individual atoms or residues in a protein structure to resolve this protein structural challenge by NMR spectroscopy. So in normal solution NMR or solid state NMR, you would try to measure all the distances between all the carbons in this kind of core, for example. But since all of them overlap in these peaks, we can't really do that. So what we had to do is rely actually on doing these kind of angle measurements by solid state NMR. And this is briefly summarized here from a paper now already a couple of years old in PNAS. And I touched upon this in more detail in this seminar that I gave in a different webinar series, which is available online, so people can visit it there if you want to hear more about it. But the basic idea is we can see different NMR signals from different structures in terms of the angles, and from that deduce the specific angles we see for the 2 conformers we see in the PolyQ structure. And with that, we can actually then resolve big part of the PolyQ amyloid conformation, and we can even apply that to the side chains. And so in this paper, we were able to show that the side chains form this interdigitating kind of conformation as reported in, for instance, the nice work by David Eisenberg in his group. So that's pictured here. So you see these side chains interdigitating in each other, and that's all largely informed by these kind of angle constraints we were able to measure with solid state NMR method measurements of these kind of angles. And a side effect of this is that we can understand that these angles fit with this idea that we know that these fibers are anti-parallel beta sheets. So most amyloids people study are parallel beta sheets where one protein stacks onto an identical copy of itself in a parallel fashion. But in PolyQ, you have that each protein contributes an anti-parallel beta strand interaction, which makes one strand different from the other, and they alternate in the high order beta sheet structure. So you get, and what we could understand here is that you couldn't actually fit 2 of the red or 2 of the blue types of structures to fit against each other. So that's, again, we reported that back then. And what I mentioned already is that in these kind of NMR spectra, we couldn't see a big part of the structure of the protein. And we could combine that with these kind of solution type NMR spectra. So inept-based NMR spectroscopy where you cannot see any signals from the rigid part of the protein, but you do see those parts of the protein that are highly flexible. And you can see here that you get quite a lot of very narrow peaks, actually narrower than we get for most of the PolyQ core structure. And these are the parts of the fiber that are on the surface. So we can now see the surface of the fiber as well as the core of the fiber in the same type of spectroscopy. And we wrote a review about how you can apply and understand these methods. We published in the journal Methods, and that's the paper that some of you may have heard Dylan reference earlier. But these are tools that people have applied in many different labs, and I especially also would note that Ansgar Siemer and his group, who study these same proteins, have published a lot of work on the proline-rich domain that's on the surface of these kind of fibers, finding very similar results as to what we see here. Now, I want to return now to kind of point out that these pictures I'm showing here, these structures of the PolyQ and Huntington fibers, this is the result of really nice work by my former PhD student, Jennifer Boats, whose most latest paper was published last year in the Journal of Molecular Biology, where we presented these structural models in which she integrated X-ray, EM, and solid-state NMR data to try to build the first, I think, fairly realistic model of what we think the PolyQ and Huntington structure looks like in these fibers, noting that they kind of appear like a symmetrical or cylindrical fibers with an equal length and width of the fiber core, allowing her to put together this idea that we must have something like 9 beta sheets all sticking together into a core, explaining the fact that we see a large part of the glutamines being extremely rigid and away from the solvent of the water in the sample. And we and other groups, including the group of Mark Baldus, have published on this feature of PolyQ based on solid-state NMR measurements of the distance between the glutamines and the water in the sample. So, but this now gives us, I think, for the first time, really kind of a reasonably good molecular model of what these fibers look like, and we can start to try and understand the changes between different fibers that we can get, polymorphs. And that's what these pictures are supposed to show, that we believe to see that we can get these narrow fibers containing 1 filament of PolyQ, and also wider fibers which contain 2 filaments sticking together in some way, seemingly mediated by these flanking regions. And you can see that here, that you see and in other cases the same different types of fibers we can make under reproducible conditions and see by antibody assays and in seeding assays, and to some extent in toxicity assays, that they are differently speaking from a biological point of view. Now, so this is kind of an interesting aspect of polymorphism. Graph where you plot the N of Huntington or PolyQ proteins, and the peaks always seem to be in the same place. This leads us to the idea that the structure I've just shown you, these atomic structures defined by these angles that we measured, must be true for all of these kind of PolyQ aggregates we have studied so far. And that in all these cases, we have this pattern of alternating blue and red arrows, blue and red beta strands that co-assemble into a fiber core. Now, the interesting thing that we have identified, though, is that if you have short PolyQ, you can get each arrow to be formed, each strand formed by 1 peptide or 1 protein, whereas for the long PolyQ, we see an intra-protein interaction that's consistent with a beta hairpin conformation shown here. So you have actually the same beta strand structure, but the long PolyQ has this additional loop that connects it. So you have both the 1 and the second conformation present in 1 protein. And this is not just a kind of interesting structural feature, but it also connects back to the work of Ron Wetzel that I mentioned before, who's done these beautiful mechanistic or kinetic studies on PolyQ aggregation, and shows that you switch to a different nucleation mechanism for short and long PolyQ. And already he really pushed for the idea that these must be somehow related to the formation of beta hairpins, and that this nucleation event of forming a beta hairpin is what facilitates the really rapid accelerated aggregation of long PolyQ, whereas short PolyQ can aggregate if you push it, but it really requires high concentrations because it needs to form a multimeric nucleus. And this then leads to kind of this idea that this beta hairpin structure that we see in our NMR studies relates to this nucleation event, and that this beta hairpin formation is what enables pathogenic aggregation. So you need this beta hairpin to get toxic effects. So the short PolyQ of a wild type Huntington, for example, could aggregate if you really pushed it, but in a functional cell, you get chaperones that counterbalance these aggregation kinetics. But in the case of the long PolyQ, now you can form these beta hairpin structures, and these beta hairpin structures are sort of too proactive in aggregating for the chaperones to keep up. So now you have a switch to toxic conditions where you get aggregation, and then you get the side effects I mentioned before of sequestering other proteins, disrupting membranes, clogging up nuclear pores, and all sorts of things going wrong in the cells that could then lead to toxicity, including perhaps the sequestration of chaperones. Now, one aspect of this is that if you follow this model, you might suggest that these different types of diseases that I briefly touched upon, so you have all these different types of disorders that all have PolyQ proteins involved, where the threshold, the point at which the disease happens, varies. So you have some where you need 40 Qs and some where you need 70 Qs, and we don't really know currently why that could be. But maybe that relates to the fact that in some cases beta hairpin formation is easy, and in other cases it's difficult. And that also PTMs and chaperones may modulate these propensities. And this then touches upon this kind of small advertisement I would like to make for some new work that we just published together with a lab in Texas, the lab of Lukas Joachimiak, on studying a chaperone, DNA JB8 in our case, that is predicted to exactly interact with PolyQ beta hairpins, thereby block their aggregation, and thereby be one of the most proactive chaperones that prohibits PolyQ aggregation. And we just published this in Nature Communications, where we applied solid-state NMR to understand the structure of these oligomeric chaperones. And we hope to build on this work more to further understand whether we can indeed tie together the chaperones to this beta hairpin formation process. Then I basically want to round up now by kind of summarizing what I've talked to you about today. Is that so Huntington's disease is this inherited disease where you get age-dependent onset of disease that relates to mutations in this Huntington protein, where the mutations take place in a disordered region of the protein that becomes prone to forming these inclusions when it's fragmented from the main protein. And I haven't really talked about how that might happen, but that these fragments then form inclusions, that we try to recreate some of these inclusions in vitro and then study their structure with solid-state NMR, and then connect that both to toxicity that we measured together with the group of Amalia Dolga here in Groningen actually, and the mechanism of formation, which we then hope to connect to disease onset. And this work has been made possible by funding from NIH, NIGMS predominantly. And currently we're also being founded by the CHDI and a Dutch campaign team that's funding one of my PhD students. Currently in the Netherlands, in my group, we have several people working on PolyQ or PolyQ related projects, including postdoc Irina Matlohov, Grishma Jain funded by the campaign team, and Raffaella Parlato funded by some university funds. In the States, I particularly want to point out the great work done by Jennifer Boats, Kyle Lin, and Cody Hoop in recent years on characterizing the structures of these aggregates. And of course, we do this with the collaborations with a number of different groups, including that of Amalia Dolga, with whom we're doing these toxicity assays here in Groningen. And I'll leave it here, and I'll welcome any questions that people might have.
Thank you very much, Patrick, for a very clear and interesting presentation. We have 10 minutes for discussion now, for questions. So if you have any questions, please type them in the Q&A. I will start with reading the first question from Alexander Kaibul. Do the double filaments fall apart when you incubate them under conditions where the single filaments are forming?
Well, so that's an interesting question. Trying to jump back to my slide. So one of the things for this paper, these results on this slide are mostly from 2017, from this manuscript. So people that want to read back on it, we made the two different fibril forms, and we included these seeding assays. And what we see there, however, is that when we seed with the wider fibers, we don't necessarily get the narrower fibers. So that means that the conditions in that case seem to dictate the morphology more than the seeding. So I think that partially answers the underlying question. I don't think we have really tested whether just incubating the fibers by themselves would lead to that kind of restructuring. There is a paper on bioRxiv now by Ansgar's group that reports that certain solvents are able to restructure their fibers into these kind of different states. So they see an interconversion between fibrillar states in the presence of some organic solvents. So I think that hopefully partially answers at least.
Okay, thank you. Then there's a question by Christian Griesinger. You mentioned that the beta hairpin is necessary for the toxicity. Is this just because then the aggregation is faster and therefore more aggregated protein is formed because the hairpin could be the seed for further aggregation, while in the shorter terms you would need two molecules to come together? You want me to read it again?
Yeah. Okay.
Oh, you got it. Here is an on-pathway intermediate during amyloidogenesis.
Well, it depends on what you're talking about. For these exon 1 fragments, they do, but that's dependent on the presence of this N17 fragment. For the PolyQ per se, there is some debate in the literature, and I'm on board with Ron's data that suggests that PolyQ per se may not be that good at making on-pathway or otherwise oligomers during amyloid formation. And that maybe is something to discuss at the end in more detail if people are interested in this. But I would say for the Huntington exon 1, it is, because that's how the aggregation kinetics are enhanced by N17, is by either making oligomers. In the case of higher concentrations or PolyQ by itself, not necessarily.
Okay, then we go to the next question. Devarajan Tirumalai, maybe I mispronounce it. Is there any indication that in isolation, monomeric PolyQ exhibits signatures of hairpin structure, even at low population level? If not, at what stage do you observe hairpin structures?
Yeah, so there's experimentally, I'm not aware of any evidence for a beta hairpin conformation being detectable, and people have tried quite extensively. So the population, if present, is very, very low. In simulations, this is different. People have reported simulations that show hairpin structures, but I think, yeah, I think that's not reflecting reality. So I think that these hairpin conformations are very rarely formed, but once they form, they're extremely aggregation prone, and they drive the recruitment of other monomers, and that's how you get the process to take place. So that would be my answer, that the monomer beta hairpins are extremely uncommon. The one caveat for that is there is some work that uses antibodies as a proposed detection of beta hairpins, but I think it's unclear that those antibodies actually really see detect beta hairpins, and that's a point of discussion as well.
Okay, next question is from Bin Xu. Very interesting talk. can you discuss further about the post-translational modification of the protein and the significance of PTMs in terms of aggregation, toxicity, and disease?
Yeah, so one of the, there are lots of different aspects there. One thing is that people have shown that the PTMs present in the flanking domains in the N17 segment reduce toxicity. So there's a mouse model where you see an elimination of toxicity and aggregation by putting in PTM mimicking mutations. And that we have probed in previously published work, and it seems to be related to destabilizing the stability of oligomers and fibers. What I touched upon at the end, which is maybe what the question is about, is that for certain PolyQ proteins, people have shown that if you have opposite charges that are related or controlled by PTMs, you can actually make the aggregation higher or lower. And I think that that could be explained by having the charges either stabilize or destabilize beta hairpin formation. This is particularly true for the androgen receptor that causes one of the PolyQ diseases, where people see if you introduce charges that bring the two termini together, you get more aggregation. So that would fit beautifully with the beta hairpin idea. So that you can see the two effects happening, that it could either increase aggregation and toxicity, or it could decrease it.
Okay, one more question. Anonymous, you showed polymorphs of amyloid fibrils. What do you think about polymorphs of oligomers and stability of different types of amyloid fibrils and oligomers?
Yeah, so we haven't looked in detail at the oligomers yet. So I don't know. I mean, the data that I'm aware of that's maybe worth noting is that for the PolyQ oligomers or PolyQ Huntington protein oligomers, it's not clear that that are containing much beta strand structure. So they're more likely to be alpha helical oligomers. And regarding the relative stability of oligomers, it's not easy to get highly stable oligomers out of PolyQ proteins. So I think they're relatively unstable. But comparing them, I'm not sure. For the stabilities of the PolyQ aggregates, we haven't measured ourselves yet. And so that's still an open question as well. Once we make these fibers, they appear to be extremely stable. I mean, they last for a really long time. And we don't see detectable changes in the NMR spectra, for example, even after long measurement times.
Yeah. There are still a few more questions, but I think in view of the time, maybe we should stop here. And Patrick, maybe you can sort of type answers or come back to these other questions at the session at the end of the ZOOMinar. Yep. So thanks again, and then I will give the word to the next chair.
Yeah. Okay, Rams, my turn.
Yeah, your turn. Yeah. So
okay. Yeah.
Can
yeah,
you stop this screen sharing, Patrick? Can you stop sharing, and Gareth can start sharing the screen.
Okay, great.
Yes, get started. Yeah.
Okay.
Yes, Rams and Pikash, thank you for the opportunity. I'm delighted to introduce Gareth Morgan, my colleague across the street from the Amyloidosis Treatment and Research Center at the Boston University School of Medicine. Gareth is a superbly trained biophysicist and structural biologist and medicinal chemist who brings an important research component to our clinical center. He has a stellar pedigree, having worked with Jeff Kelly at Scripps and Sheena Radford at Leeds, among many other superb labs. And early in his career, Gareth developed a lifelong interest in protein structure, stability, folding, aggregation, amyloid formation, and he worked on a number of various amyloidogenic proteins. So originally, Gareth comes from UK, where he completed his undergrad from Imperial College. Then he received a PhD at the University of Sheffield, and that's where he worked on NMR structural studies of amyloid, of cystatin. From there, he moved on as a postdoc, first to Leeds, where he worked with Shane Redford on protein evolution, stability, and function links between these aspects of proteins. And then he moved from there to San Diego, California, where he worked for a number of years with Jeff Kelly and honed his skills in medicinal chemistry, worked on TTR, and started working on light chain amyloidosis, and developed this approach where he is trying to stabilize the natively folded protein precursors of amyloid to protect the protein from misfolding. And that's what Gareth is going to talk to us today. Take it over, Gareth.
Well, thank you very much for the kind introduction, and thank you for the invitation. It's great to be here to talk to you. I'd like to talk today about something a little different from what Patrick has just told us. I'd like to talk about how we can go about designing drugs to prevent amyloid disease and the progress that we've been able to make in designing drugs for a particular disease, which is called light chain amyloidosis. So this has been a long-standing collaboration with Jeff Kelly at the Scripps Research Institute. And I should point out before we start that I do have something of a commercial conflict of interest with this. We do have a patent on the molecules that I'm going to show you today, and we're looking to develop these into a drug with the help of a commercial company. We can talk a little bit about the ethics and the practicalities of that in the discussion if anyone's interested. So unlike in the case of Huntington, where and other amyloid diseases, where the precise role and the nature of how amyloid fibrils cause toxicity is not really well understood, in some diseases there is really clear evidence that accumulation of these amyloid fibrils actually causes pathology. And in particular, in a class of diseases called systemic amyloidosis, an accumulation of amyloid in the heart is extremely problematic. So this is an explanted heart from a patient with light chain amyloidosis. And I'm not a cardiologist, but hearts are not really supposed to look like that. This big, thick wall of muscle on the center and right of the heart is not healthy. So even in the absence of any kind of specific mechanisms of cellular toxicity, then it seems as though just accumulation of amyloid fibrils in the spaces between cells, particularly in tissues such as the heart, but also in the kidney and elsewhere, can cause problems. So there is also known to be a series of mechanisms by which light chains and the precursor proteins and amyloid fibrils can cause acute toxicity on top of this kind of bulk displacement of tissue, but those are less well understood. So in the disease that I'm going to talk about, this is called AL amyloidosis, or light chain amyloidosis, and this is caused by the light chains of monoclonal antibodies. So the light chains I'm showing in this cartoon representation, which I'm going to use throughout, and at the top here, are essentially fragments of mature antibodies, which is what you might be more familiar with, the structure of an IgG antibody down below, where you have a variable domain and a constant domain. The variable domain binds to antigens, and the constant domain has a scaffolding and chaperone function. And these light chains are released alongside full-length antibodies by B cells and the plasma cells that are specialized antibody secreting cells in the bone marrow. So for those of you with a more structural disposition, then this is this is a more detailed view of this same structure. So we have 2 beta sandwich immunoglobulin domains per chain, variable and constant. Those are linked together into a dimer, and that dimer can be stabilized by an intermolecular disulfide bond, which I'm showing as a black line here. So in AL amyloidosis, then these free light chains are secreted by monoclonal plasma cells. These are specialized antibody secreting B cells that mainly reside in the bone marrow. And what you see is in healthy people, then we have millions of different plasma cells secreting millions of different antibodies. But in amyloidosis, then you see one specific plasma cell clone that comes to dominate the antibody repertoire, and it's that one clone that seems to produce the light chains that aggregate. So these light chains seem to be able to cause toxicity on their own. There are a number of studies looking into that, but they can also form amyloid fibrils, and the combination of these 2 events really leads to progressive organ failure. And patients have a very poor prognosis unless they're diagnosed early in the disease. It's possible to treat the disease. Treatments have actually advanced quite a lot in the last couple of decades. In particular, the way that this is treated is to use cytotoxic chemotherapy, originally designed for multiple myeloma, which is kind of a more aggressive form of the same cancer. Using this cytotoxic chemotherapy to kill the precursor plasma cells and therefore reduce circulating levels of antibody light chain below the critical concentration needed for amyloid formation. For a long time, there have been efforts to use antibodies against amyloid or antibodies against an amyloid-associated protein, serum amyloid P component, as a way to direct the immune system to clear pre-existing amyloid deposits. That's kind of been on the cusp of being successful for a long time, but it's not yet shown really good efficacy in the clinic, and it's turned out to be much more difficult than was originally imagined to be able to remove pre-existing amyloid fibrils. What I want to talk to you about today is a different strategy that we think is complementary and potentially useful in addition to these approaches, which is to stabilize the precursor light chain using a small molecule that binds to the native state of the light chain and prevents it from misfolding and aggregating. So how do you go from a folded light chain to an amyloid fibril? Well, we know that there must be multiple steps involved, and we know that there must be significant conformational changes because the structure of the native light chain is completely lost in the structure of the fibril, and you can kind of see this with the topologies of the 3 known light chain amyloid fibrils that I'm showing on the right here. There seem to be alternative amyloid fibril structures, but in order to form them, you need to lose the native structure of the protein, then you need some kind of series of self-assembly steps in order to bring the monomeric proteins together and have them aggregate. In the case of light chains, then there also seems to be proteolysis because the core of the fibrils is primarily composed of the variable domain, whereas the secreted species, it also includes the constant domain, and somewhere along the line that seems to be lost. So we know that unfolding is important, and that means that when you study this, the kind of, if you set up a typical amyloidogenesis reaction and look to see what the kinetics of amyloid formation are from the folded proteins, then the rate at which these proteins form amyloid is really dominated by the stability of the native state of the protein. So this is some of our work, although these similar results have been seen by multiple people using different light chain systems. So this was one we published a couple of years ago. We used NMR hydrogen exchange to calculate the stability, which is the proportion folded, which is on the x-axis there, and measured the rate at which the proteins formed amyloid fibrils by thioflavin T kinetics. And we saw a reasonably good correlation at a single concentration of light chains between multiple light chains, all from the same original light chain precursor gene. So there are the two colored points there named JTO and WIL are two patient-derived light chains, which are all related to the wild-type protein, which is in pink. And then we made a series of point mutations to try and investigate the mechanisms by which these proteins form amyloid. So we can come up with this relatively simple scheme by which unfolding precedes some kind of self-assembly. So you need to be able to form an amyloidogenic intermediate, and this is going to be some fraction of the unfolded or partly folded ensemble. And these amyloidogenic intermediates can self-assemble into initially small oligomeric structures, which will eventually grow into amyloid fibrils. In the case of light chains, as I mentioned, then proteolysis seems to be involved, and it may be necessary for initiation of aggregation that some part of the protein is cleaved by proteolysis to leave an amyloidogenic fragment. Now we can, if we want to study these processes, we can kind of isolate different steps, and we can measure different rate constants and calculate equilibrium constants between all of these different species. But it can really be a challenge to successfully isolate the one particular process that you're really interested in studying. However, because we have all of these different processes, then what we hope is that there are multiple places around this pathway where you can potentially intervene with a drug of some description and potentially inhibit a process that would normally lead to aggregation. So obviously on the left we have the cytotoxic drugs which suppress formation, but you can potentially envisage molecules that inhibit various different steps of the amyloidogenesis cascade. And the important question is, what are the best targets for intervention, and how do we go from a molecule that inhibits this a little bit in a test tube to a molecule that is really effective at inhibiting these processes in a patient? So when you look at the process of amyloid formation after a protein has unfolded, then you can kind of imagine it on this very simplified energy landscape where there is a transition between the unfolded state and various different potential amyloid fibrils, which is associated with the self-association reaction. In the case where there are already pre-existing fibrils, then extension seems to be easier than initiation in many cases, and this is where you get these kinds of seeding effects. And you can envision that as reducing the energy barrier to aggregation. And it seems that although there are multiple different fibril forms possible for any particular sequence, the energy barriers in between them are quite high. So you're probably not going to see conversion between different amyloid species without going out via an unfolded state. So there are lots of ways that you could imagine a small molecule could interfere on this pathway. You could, for example, you can stabilize non-productive species, either monomers or aggregates, and kind of direct the flux of the reaction towards off-pathway species. You can block self-association reactions, whether in terms of blocking the interaction between two monomers, or blocking the interaction between a monomer and a pre-existing fibril. So the fibril capping oligopeptide reagents that David Eisenberg's lab has done quite a lot of work on, for example, would sort of fit into this scheme here. And in order to measure that, then typically people use thioflavin T, which is this heterobicyclic dye here. Thioflavin T can bind to amyloid fibrils. It can also bind to various other things. And when it binds, the rotation between the two ring systems is fixed, and that increases its fluorescence. And so you see an increase in fluorescence when you bind amyloid to thioflavin T. And so you can imagine using this, and in fact, many people have used this as a way to screen for molecules that inhibit amyloid formation. So you can take a reaction where your thioflavin T signal increases, and you can add your small molecule to your aggregation reaction, and you can look for ones where the thioflavin T signal does not increase. Unfortunately, thioflavin T is prone to false positives and false negatives. In the case of where you're screening for a potential anti-amyloid drug, then false positive hits are a particular problem. So you can see displacement of thioflavin T by binding of the small molecule into the similar site. Or you could potentially see quenching of thioflavin T's fluorescence by your small molecule. So as a way to kind of look into this, then we actually, this is a simple experiment that is part of a slightly larger investigation into a number of small molecules that have been shown to interfere with amyloid formation in various different systems. So what we did here was we took light chains and added thioflavin T, and we aggregated them for 24 hours until we formed fibrils. And all of these reactions formed fibrils, as we would expect. But then we added the small molecule and then immediately measured the fluorescence emission again. And what I'm showing here is the change in fluorescence emission upon addition of small molecule. So in this case, we don't expect that what we're seeing here is complete dissolution of the amyloid fibrils, and in fact we know that we don't get dissolution of the amyloid fibrils. What we're seeing here is some sort of artifactual quenching or displacement of the thioflavin T that leads to suppression of its fluorescence. We have a preprint of this manuscript up, and there should be a final version of it before too long. So if self-assembly and amyloidogenesis itself are difficult to target by small molecules, then we think that a better way to go is actually to target the native antibody light chain itself. And so this is what leads us down the path towards native state stabilizers. So there are two advantages. The first is that you really know a lot more about the structure that you're attempting to target. We know the structures of the native state, and we're able to work quite well with the native state, and we know how much of it is in solution at any given time, which is not really the case with all of these transient intermediates. And also because there are relatively fewer moving parts in the kind of assays that you'd want to use for this, then it's easier to identify different ways in which there are small molecules, that your small molecule is actually working. So this is not a new idea. This is basically an extension of what has been done with another common type of systemic amyloidosis, which is transthyretin amyloidosis. And Jeff Kelly's lab and various others have shown that small molecule binding to the native state of transthyretin, which is a native tetramer, stabilizes the native tetramer and reduces the rate at which the tetramer dissociates into amyloidogenic monomers, which are the species that can actually go on to self-assemble into the fibrils. So this has been shown using a design drug, tafamidis. It's also been shown using several other pre-existing drugs in a kind of drug repurposing study. So tafamidis was recently approved in the US for amyloid cardiomyopathy, transthyretin amyloid cardiomyopathy. So in terms of trying to find a known structure, then for transthyretin, the transthyretin has a binding pocket. It binds the thyroxine T4 hormone, and so designing a molecule that will bind to transthyretin is kind of a bit like spearfishing, where you can basically choose the fish that you want to target. In some cases, you can do this with a lot of small molecules, particularly if you know the structure and there's a known binding site. With light chain, this is not the case. An alternative approach, which also worked for transthyretin, is kind of to repurpose an existing drug. And so continuing my fishing metaphor here, then it's kind of, you don't necessarily know what you're going for, but you have a pretty good idea that what you're trying to do will work in the same way. Drug repurposing has been in the news quite a lot over the last year, with rather limited success in the case of hydroxychloroquine, for example. But if you can make it work, then this can be really, really effective. And actually, transthyretin stabilization is one of the sort of standout successes of this drug repurposing process. And diflunisal, which is a small molecule non-steroidal anti-inflammatory drug, a painkiller, has been used quite widely as a stabilizer of transthyretin. But if you don't have any of that information, then what you need to do is basically go for a high throughput screen where you cast as wide a net as possible and you pull in as many things as you can that will hit your target protein using some sort of high throughput screening assay. So a good example of this in terms of protein stabilization is the small molecules that stabilize the cystic fibrosis transmembrane conductance regulator, which were developed by Vertex over the last 20 years. This was a huge amount of effort to do this, but the resulting drugs have really been transformative. So, as I said, we don't know a binding site for ligands on light chain. We don't really know whereabouts you would be able to bind something to light chain in order to stabilize it, and we don't know what such ligands would look like. So that left us with high throughput screening as our way forward. And so those of you who've done CD or fluorescence urea titrations, for example, will know that measuring stability is a reasonably challenging and painstaking process. It's not suitable for high throughput screening, so this is the method that we came up with in order to screen a large library of compounds for potential stabilizers of light chains. So we're going to use the observation that proteases will preferentially cleave unfolded peptides. So if your protein is folded, then it's only cleaved by a protease when it transiently visits an unfolded state, and it's that process that we're looking to inhibit. So to measure that in a high throughput way, we labeled our proteins with fluorescein, and then we measured the change in fluorescence polarization as we degraded the protein with a protease, in this case just a low specificity protease, proteinase K. And as the protease degrades the light chain, then you see release of smaller peptides which have a much faster rotational tumbling time, and therefore a low fluorescence polarization. And so the idea is that a stabilizer molecule will reduce the rate at which the light chain is degraded by the protease, and therefore reduce the change in fluorescence polarization. So this works reasonably well, surprisingly well almost. We were able to miniaturize this to 1536 well plates, and then we sent it to the Scripps, Florida high throughput screening facility, which screened it against 650,000 compounds in their library. and we were able to find some hits. So we were able to show that the small molecules that, firstly, the small molecules that were able to be active in this screen and then maintained activity when we used multiple counter screens to eliminate false positives. We showed that these molecules are able to bind to the light chain. This is ITC showing binding to unlabeled light chains. We showed that addition of the small molecules increases the amount of urea that's necessary for the protein to unfold. This is measuring intrinsic tryptophan fluorescence. And we showed that using a non-obligate dimer, which is the red traces here. This is analytical ultracentrifugation. Addition of a small molecule to the non-covalent dimers shifts the monomer dimer equilibrium towards dimer. So it looks as though these molecules are inducing dimerization. So this is the molecule that we've focused most of our attention on, partly because we already had a bottle of it in one of the cabinets in Jeff's lab. This is coumarin 1. It's a known dye. It's relatively cheap. The coumarin is shown in black, and this is a diethylamino 4-methylcoumarin, and the additions are shown in red. So coumarins are known fluorophores. They're also known to be known as molecules that often generate false positives in fluorescence assays. But we're able to validate this using non-fluorescent assays and show that it really does work. One thing that I've learned over the last couple of years is that medicinal chemists really don't like coumarins, and there are some metabolic and toxic liabilities associated with the coumarin that we kind of want to avoid. But critically, this was a really useful tool compound for us. It allowed us to learn an awful lot about what's really going on and how these molecules stabilize the light chains. And a big part of this reason is that, as I mentioned, coumarins are dyes, they're fluorescent, and this one in particular, coumarin 1, is an environment-sensitive fluorophore, which increases in fluorescence when it binds to the light chain. So we can basically measure binding between the small molecule and the light chain just using the fluorescence of the small molecule. You can see there's this blue shift in the red trace here. And we can take advantage of this to titrate the protein into the small molecule, and we see binding to the full length light chain, binding to its variable domain, but no binding to its constant domain. And the poor fit of the red traces is due to induced dimerization of the variable domain when the small molecule binds. So at this point, a talented graduate student called Nick Yan joined the group, and along with David Mortensen, who's a crystallographer in Jeff's lab, we were able to solve the structures of the light chain both on its own and bound to our small molecule. So this is the structure of a full-length light chain with the small molecule bound, and you can see it binds in between the two variable domains at the interface between them. So if we look closer, then we can see that there's this kind of pocket made of hydrophobic aromatic proline residues that's in between the two chains of the dimer. And what's really encouraging about this, and what was really very exciting, was these residues that form the binding site also form the interface between the light chain and the heavy chain in a regular antibody. And so these are really highly conserved among multiple different light chains. And when every individual patient has their own unique light chain, we really need to find a conserved binding site in order to have a hope of being able to design a drug that will work on multiple different patients. And so this conservation was really encouraging. However, One thing I will point out is that coumarin-1 is quite a flat planar molecule, and it kind of slides into this binding site. One other class of hits that we found from our screen is a series of molecules containing a hydantoin group. And these are not planar molecules, these are distinctly curved. So you can see coumarin-1 is relatively flat, and compound 8 here is decidedly not flat. And so the question that we asked is, how does a curved molecule fit into a flat binding site? So to answer this, we first turned to computational docking, reached out to Stefano Folli's group at Scripps, and Diogo Santos Martins, along with Nick, was able to simulate the docking of small molecules into the crystal structure of the bound light chain. So you remove the existing small molecule from the electron density and use autodock Wiener to do a sort of rigid body docking with your molecule. So we first showed that you can dock the original hit, the coumarin, into its own binding site, which is useful and encouraging. But then we also showed that these hydantoin molecules, they fit into the same binding site, but they actually stick out of the binding site and really extend the interaction surface between the small molecule and the light chain. And this is really interesting for us because this allows us to potentially build larger molecules that will enhance the affinity between the small molecule and the light chain. So Nick was able to solve the crystal structure of this, of a similar compound, it's not quite the same compound, and showed that it binds in the orientation that was predicted by the docking algorithm, and used that to try and identify residues in the light chain that we could potentially target for further medicinal chemistry development. So when we want to, if we want to make an actual molecule that is going to go into patients and be, you know, a potential drug candidate, we need it to have a number of things, and we really want to optimize as many of these factors as we possibly can before we can start thinking about human or really even animal trials. So what we're doing right now is trying to generate the best molecules we can, firstly by optimizing affinity and selectivity, but then they also need to be soluble, bioavailable. They need to survive in the body for a reasonably long time and not be metabolized to something that's toxic, and indeed just not be toxic themselves. And so this process of sort of designing new molecules that are more efficacious and more drug-like is going to take us quite some time. But what I'd like to just finish with is talking about how we've made our first steps towards high affinity compounds that are more selective for light chains. So this was our hit molecule, and we kind of divided it into an anchor region and a core region. So we kind of think of the coumarin as the core that binds, that makes this pi stacking interaction you can see in the structure, and then the anchor kind of holds it in place. That's buried deepest into the protein. So Nick was able to extend this idea based on what we'd seen in the crystal structures with the hydantoin, by extending the coumarin out in the same direction as the hydantoin kind of points to make similar kinds of interactions with these additional residues outside our original core. And so we kind of classify these as having a linker region and a distal region. It's kind of a four substructure design system. And so Nick along with his colleague Reggie Nair has synthesized and purified and tested around 250 small molecules at this point, really initially trying to individually optimize the distinct parts of the light of the molecule, and then combine those together to make a kind of best-in-class molecule. So this is an example of one of the compounds that was made to optimize the linker region. You can see this green, I think this is a spirohydantoin moiety that's been added to the coumarin. And this is our first molecule that has sub-micromolar binding affinity, or at least what I'm showing here is EC50, which is the effective concentration at 50% inhibition. And in this case, inhibition means inhibition of proteolysis of the protein. So it's essentially 50% protection at this concentration of light chain in our screening assay. So Nick also made a series of compounds to kind of optimize this distal region, which makes interactions which are now quite a long way removed from the original binding site, but still with relatively conserved residues at the interface between the two light chains. So this molecule now has an EC50 of 77 nanomolar. And then finally, we're able to take our best linker and distal region and combine it with a more optimized anchor substructure, in this case, changing from the original coumarin to a phenylethoxy coumarin here. And this molecule actually now has an EC50 of around 2 nanomolar, which is comparable to the affinity of tafamidis for transthyretin. So this is much more in the range of being a drug-like affinity, which is what we're really looking for. And we don't have the crystal structure of this yet, but this is a docking model of the same structure, and you can see that this phenyl group is buried deeply within the protein. So this is the progress that we've made. This is, as I said, the first of several steps towards taking a small molecule from going from a screening hit to an actual drug-like lead molecule. We have many more things to optimize on this. There's a long way to go before we get to patients. And critically, it's really important to think that, there are very few good animal models of systemic amyloidosis, and none of them recapitulate all of the facets of the disease. So it's really important to try and get a molecule that's as good as possible in vitro, because the only way that we're really going to be able to ask whether our hypothesis that stabilizing the light chains will reduce amyloid formation and benefit patients is to actually do the human clinical trial in patients, and to ask, to look to see whether this really makes a difference. And so that's clearly still a long way off, but we're making progress towards this. And this paper was published just a couple of weeks ago. And I'm so far cautiously optimistic about this. So I'm going to wrap up there. I think I've kind of told you how, there are multiple processes that you could potentially target for intervention. We aimed to target a relatively early state in this amyloidogenesis cascade, because we know much more about what's going on with the native antibody light chain than we do about any of the intermediates, which, the things that we see in vitro may or may not be the important things in patients. And so stabilizing the native state seems to be critical. But what's nice about this model is that you could potentially also do some of the other, take other approaches and potentially combine multiple different approaches to yield a sort of synergistic benefit for patients. So this work has mostly been done at Scripps Research. There are a number of people who have contributed to this, most particularly Jeff Kelly, who provided the funding and allowed me to sort of wander off on my own until I came out with this idea of a testable hypothesis for stabilizing light chains. And Nick Yan, who was a graduate student in Jeff's lab, who's done a huge amount of the structural biology and medicinal chemistry. Since then, I've moved to Boston University to join the Amyloidosis Center, which is a translational research center where we combine both, a clinical center of excellence. We work quite closely with physicians, and we also do basic research and also kind of clinical diagnostics. And so our work here is not quite ready for public publication yet, but there's a great team, and it's been a real honor and a privilege to join this group of people. So I will stop there. Thank you very much for your attention.
Thank you very much, Gareth, for a fantastic talk. Let's have questions. If you would like to ask a question, use the raise hand function in the Zoom, and I can enable you to ask the question directly to the speaker. Questions from the panel? To speak.
I have a question.
Yeah, go ahead, Bikash. Go ahead.
I have a question for Patrick. Like I was very fascinated about your solid state mass and solution state mass result. I was wondering, like when you are looking at the flexible regions using the solution state mass, have you ever compared those spectra with the monomers or the oligomers? How do they differ? And the second thing is that, are they very polymorphic? Because you mentioned that your fibers are not that polymorphic, but how about those flexible regions?
I can answer, but I wasn't sure if we should first do the questions to Gareth's talk. I don't know what.
Yeah, so let's see here, the Q&A has any questions.
There's one from Alexander.
Alex, would you like to join us in the, let me see here. Alex, go ahead, ask your question. Alexander Buell.
Hello, can you hear me?
Yeah.
Okay. Okay, so yeah, I'm impromptu live here. Sorry. Yeah, my question was, what is your guess about what's the difference between amyloidogenic or amyloid-forming light chains and the light chains that deposit amorphously in patients, which lead to kidney damage and in other ways? Like that seems to be more frequent that it's not amyloid-forming. So what do you think is the difference?
So the difference between light chains that damage organs by forming amyloid and light chains that form that damage organs.
Yes. So basically,
the answer is that we really don't know.
Yeah.
Yeah. So I think there seem to be there are a number of kind of places where light chain amyloidosis is extremely diverse. One is that the individual has their own light chain sequence, and it's produced at slightly different levels, and each individual has light chain deposition in different places, and that can have different consequences. And it's really not clear what's driving all of those differences. The sequence of the light chain itself, which is a big part of it, or whether there are differences between individual patients, other proteins and other factors that are affecting that. And it seems as though these in some ways the non-native structures seem to be important, but clearly there are multiple different non-native structures, and they can form multiple different interactions with different tissues. But trying to untangle all of those moving parts has so far proved extremely difficult. And so we don't have a really good sense of quantitative factors that really drive those interactions are.
Okay, thanks.
Okay, a question from Caesar. You can ask directly. You are on the panel now. Hello, Caesar. Okay, I can read the question.
Hello.
Oh, that's for Patrick. Okay, so let's say.
Yeah. No, this
Go ahead,
was
why
a
don't
discussion
we-
with Patrick.
Go ahead.
Sorry.
Let's take all the questions, Patrick. Go ahead. Caesar, go ahead.
Okay. Thank you. I was talking with Patrick. How does he measure the fibrils for the cytotoxicity assays? Because we usually estimate the amount of fibril from the amount of monomer that we usually use to start the aggregation. But then sometimes we get challenged by the reviewers, saying that perhaps you don't get 100% aggregation from that amount of monomers, and also that you can also lose some fibrils during the washes for the sample preparation. And indeed, I think for us it is very difficult to estimate the amount of fibrils, because once you have the fibril, you cannot use some traditional ways to measure protein. And also, if you have a very stable fibril, it's very difficult to disassemble it, just to get back the monomers and use traditional methods to measure the fibrils. So this is one thing that I was asking Patrick in the question and answer. Thank you.
Yeah, I mean, all I can say is that I think I agree that it's not easy. And then one of the main things that I think how we try to work around this is to focus perhaps more on comparisons between polymorphs than to try and measure an absolute toxicity, because that would be really tough to know for sure. But yeah, I don't know that I'm not aware there is a super easy and quantitative way to know for sure how much protein and fibers you have. Not to mention the question of, like, are 1,000 long fibers the same as 10,000 short fibers? And I mean, like, it gets really complicated. I mean, we do more and more EM analysis these days, and you start to look at the complexity of the samples you have, and it's really difficult to know what kind of numbers you put on something. And it raises questions about published assays of toxicity, where people, I think, often have no idea what they're exposing their cells to, and they report some toxicity, but you don't really know how much protein is there, or what state it's in, or what kind of oligomers do you have, and how reproducible is any of this. So I think it to me seems a big challenge. And if somebody has a good suggestion, then I think both Cesar and I would be happy to hear it.
Christian Grisinger.
Yeah, hello, thank you very much. I had a question to Gareth. On the stability of your molecule in, I don't know which animal you are testing it for, metabolic stability.
Yeah, so we have the animal data. What we have done is we've looked at plasma protein binding, and so this is something I sort of alluded to but didn't really go into any detail. If you have a molecule that you want active in blood, then you want that molecule to bind selectively to your target protein rather than to albumin, which is in blood at 1,000 times higher concentration. We've shown that these molecules that Nick has made bind quite tightly in plasma, and that is consistent with a long half-life in blood. Good, but it's not clear whether it's not yet clear whether we have the selectivity that will allow you to allow us to kind of actively into as much of the light chain as possible without needing, a vast excess of the small molecule.
Okay, thanks a lot.
Bikash, go ahead, ask your question.
I'd like to repeat my question again. So I think I like the solid state mass and the solution mass spectra that you are showing to compare the rigid versus the flexible part. So my question for Patrick is that, do you know how polymorphic these flexible regions are, and how do they differ from the monomers and the oligomers? And in connection to that, my next question is that people generally do digest the flexible parts by using trypsin or clip B, and they to take the core region to look at the toxicity of the fibers. So have you ever thought, because you have the solid state NMR spectra, so have you ever tried to look at whether the toxicity that you are getting for this fiber is either from the fiber core or because of the polymorphic region of the flexible part?
Well, one feature of our proteins is that they are containing PolyQ segments, but also polyproline segments, which means that there are actually trypsin cleavage doesn't do that much against either of those parts of the protein. And so you can't actually strip off the flanking domains with trypsin because there's no suitable cleavage site. So you'd have to use other ways of doing that. Yeah, we actually use this because in the paper from last year, we can use trypsin to cut our fusion protein and to release most of Huntington exon 1 and let it aggregate in absence of the N17 part of the protein. We are looking in some data on experiments at the toxicity of that version versus the complete exon 1, but that's still a bit ongoing. And yeah, if you want to do it carefully and quantitatively, you quickly run into all sorts of control experiments that become necessary that we haven't fully resolved yet. So the differences are, I think, not hugely dramatic right now, but we're still working on that. Regarding your question of the structure of the NMR or the monomer, we haven't done that yet. We're working on that, but work in a paper by Ansgar Siemer from USC, they have published solution and solid state NMR on the fibers and the monomer, and I think they show that the signals from the flexible region are very close, or perhaps virtually indistinguishable, consistent with it being an intrinsically disordered kind of conformation that's dictated by the local sequence and structure rather than interactions. That answers your questions.
Yeah, that's great. Yeah, thank you.
Gareth, there is a question for you in Q&A box from Yango Lee. I think he has left. I can read it for you. Thank you for the nice talk. Your idea is based on thermodynamics for intervention of amyloid formation. I'm not sure whether equilibrium is applicable even in our body. What do you think about this? I don't know this. Go ahead.
That's a really important point, and I'm sure that Jeff would have been horrified and actually use the term kinetic stabilizer at any point in the process today. So yes, if you look at the body, proteins are secreted, and they're either excreted or they aggregate or something else happens to them. We never reach equilibrium, but there is a state. And so in terms of trying to think about what the stability of the protein is, and rather than thermodynamic stability, it may be more useful to think about this in terms of a kinetic stability, which is the barrier between the native state and the unfolded states. You can have how rapidly, how frequently the protein folds, long ends in its native state before it kind of makes an excursion to a less folded state. So what's nice about proteolysis is that it actually, a method of measuring stability is that it looks at it actually looks at kinetic stability. And so as your protein transiently unfolds, then it becomes susceptible to proteases, which can cleave it. And so screening for stabilizers against proteolysis, we're screening for thermodynamic and for kinetic stabilizers. So you can stabilize a protein either at the thermodynamic level by reduce native state free energy versus the free energy of the thing that you want to avoid, which in the unfolded state. But you can also stabilize, but that decrease of the native state free energy, if you don't, if the small molecule doesn't bind to the transition state or the unfolded state, then you also see an increase in the kinetic barrier between folded and unfolded, and therefore you'll get kinetic stability. And so tafamidis is always a kinetic stabilizer because it's really that barrier increase function was critical for effect in vitro in humans.
There's another question from Sylvia. Sylvia, you're on the panel. Would you like to ask a question directly?
Oh, yes, of course. This question is for Gareth. I thought you did a very nice job with your talk, Gareth. And I was just wondering if there is any data so far on the toxicity of your best targets that seem to have quite a bit of aromatic rings. And so I wonder, you know, if those might interact, and also the selectivity, right? It might interact with other cellular components. And if there is any information on the toxicity, it would be great to know if you know anything about the origin of it.
So we haven't done that. Our basic strategy is to start by proving affinity, because it really wasn't clear when we set off that you could make a nanomolar binder against a protein that doesn't have a small molecule binding site, didn't have a known, well-defined small molecule binding site. So that's kind of been our priority. The fact that now that we've actually been able to demonstrate that, now the eventual molecule that will go into clinical trials, I hope, will probably look like the molecules that we have. But the key advance made here is to identify the binding site and the binding conformation of the light chain. And so you can kind of design something with drug-like properties around the binding site. So the current challenge of these molecules is that they're not very soluble. And so our next step is going to be to kind of optimize solubility to remove kind of known toxicophores or known toxic species. So that's, one of the things Jeff hates the coumarin is that there are potential metabolic liabilities in that coumarin. And so we've been trying to coumarin core alternative moieties. There's the that diethylamino group on the original is modification and conversion to toxic nitro groups in animals. And so there are lots of sort of known things that could go wrong with those structures. And so at the moment, we're holding off on doing any kind of animal work or toxicity work until we have a molecule that we're really quite confident about its ability. And we're going to ask a bunch of medicinal chemists to try and identify things that could go wrong before we really do that. So that's the strategy.
Sounds great. Thank you.
Sumei Zhang, ask your question.
Hi, my question is for Gareth, and it's kind of related with the previous question. I want to ask, how is your coumarin binding to this light chain, the specificity, and whether this binding pocket exists for all the light chain, or is specific for certain antibodies at light chain? And the other way is when you have this structure optimization, does that enhance certain specificity to certain targets?
Mhm.
And that's it.
Yeah, that's a good question. That's thank you. That's kind of what we're trying to do at BU, and we're moving towards beginning to answer. So the binding pocket is formed by a movement of the two variable domains away from each other that generates the place where the small molecule binds, and that bind site is of the residues that would normally form interactions with the antibody heavy chain. And so those are highly conserved, and we expect to see those in the vast majority of patient light chains. We don't necessarily expect to see, we wouldn't necessarily expect that the small molecule bind with similar affinity to everything. And so this is another reason to work on improving the affinity and the binding so that you have kind of binding energy to spare. So when you find a less well binding light chain, then it's still able to have some effect. It's not very specific because it binds to the coumarin hit molecule binds to albumin with a pretty similar affinity to how it binds to light chains. And so if you want to saturate your light chains in a patient, then you would need to saturate all of the albumin or a substantial amount in order to have an extra light chain to do that. And so that's why we're aiming at improving the specificity. What it is specific for is the regular head chain light chain variable domain interface in most antibodies. So which binds to the light chain dimer, but not the light chain heavy chain dimer. So that's encouraging.
Okay, thank you.
Thank you very much, Gareth. So it looks like there are no other questions in Q&A or nobody's raising their hand. Can we go back to the questions for Patrick? I know, Patrick, you answered all the questions by writing. Is it possible that you can read it out and answer them so that it can be recorded?
Oh,
There are only like 5 of them, or I don't know how many of them. 3 or 4.
I think I have to try to think. Some of them I wrote something that I had already answered. So
if you go to the Q&A and then select the answered column.
Yeah. What I'm saying is some of those I added a comment that I had already answered before. But somebody asked about saying, I was wondering if there's any more detail available on the mechanism of action of the chaperone against the aggregation of PolyQ, and does the chaperone disintegrate aggregates or constrain the conformation? And also, does the chaperone bind directly to the PolyQ domain or in vicinity, and is it specific since it's targeting a repetitive sequence of PolyQ? So questions worth probably an entire talk. But what I was going to say is that the chaperones that I mentioned, these are 2 HSP40s, DNA-JB6 and DNA-JB8, are thought to recognize the PolyQ specifically and are seemingly specific to certain kinds of amyloid forming proteins, so that touches on the specificity. They seem to bind to the PolyQ domain, and that's kind of special because other chaperones inhibit Huntington aggregation by binding to the other parts of the protein, not the PolyQ. And as such, these HSP40s are thought to bind to or sort of act on all the PolyQ proteins. And they also, what was the other question? Yeah, these 2 specific ones act more by stopping aggregation than by disaggregating existing fibers. But other HSP40s, of course, are thought to do the reverse, like they act on a fiber once it's made. So that was a question by Sunil Kumar. So I hope
There's a question from Bin Su in the top. Very interesting talk. Can you discuss further about the post-translation modification of the protein and significance of PTMs in terms of aggregation, toxicity, and disease? Did you already discuss this?
I don't remember.
Oh, maybe,
but
yeah,
I can say that what I didn't remember in the talk, I could think of is that it's the work by William Yang that really set this interest in this in motion, I think. at UCLA, and they showed really nicely that Huntington PTMs can inhibit the toxicity in mice. More recently, I think especially the work by Hilal Lachwell and his group at APFL in Switzerland have worked a lot on different PTMs and how they are really important for understanding the behavior of Huntington and its aggregation and toxicity. So that's worth looking up, and I posted some DOI codes in the chat. Most of that,
There's
as I
one
said,
from,
yeah.
Go ahead. Sorry, go ahead.
Oh yeah, there's one from
Prabhakar and Ramakrishnan.
Yeah, okay, thank you. Given the polar nature of PolyQ, how does ionic concentration affect amyloid formation? That's something that we have actually been very interested in recently in experiments. The effect is not huge in our hands so far. And what I mentioned also is that the salt concentration we cannot interpret in terms of PolyQ per se, because we are looking at Huntington exon 1, and then the N17 is probably really affected by the ionic strength, because it features both hydrophobic and electrostatic interactions that really change the kinetics of aggregation, which of course is also the mechanism by which the PTMs are relevant. So we are looking at this, and hopefully we'll have more to report on this in the future. And then somebody asked, Dr. Pratima Mishra asked about more discussion of the tomographic results. And I want to maybe mention that that's not my work. And I put the DOI code in there. That's the work by Wolfgang Baumeister and other people who applied EM tomography to cells, hex cells and neuronal cells in which Huntington was aggregating. And they showed those inclusions, and I put the DOI for the paper in there, but it's not my work specifically. And then the last question was from Cesar, which we discussed already about the concentration.
So that completes all the Q&A questions. Thank you all very much.
Wait a minute, Rams, could I ask one more question to Gareth?
Yeah, go ahead. Yes.
So Gareth, I liked your presentation very much, but the connection was not too good. And I was wondering about the experiments with the small molecules where you looked at what exactly THT is reporting. And I was wondering if you may have published this somewhere that I can look it up and read through it, because I really would like to go through it carefully.
Yes, I'm actually in the process of revising that manuscript. It kind of turned into a little story, but I will I'll send you the preprint.
Oh, thanks very much.